When you look at cells in culture, they're basically pretty much the same, just to the eye. You look at them with a simple microscope or something.
It's evolution in a dish, and these things are going to, undergo changes.
488 misidentified cell lines used in research throughout academia.
The impacts on the science, on the credibility of the science, all comes into play.
The younger generation of scientists, they are literally blown away by the problem.
We have to try and shake the cage of complacency in the scientific biomedical field.
From a day-to-day basis, we are constantly trying to look in the literature and try and reproduce data. And more often than not, we can't. If you haven't got the right cell line, or if a paper has been published using the wrong cell line or a contaminated cell line, this can affect results quite dramatically.
I think people know about cell authentication, but they don't understand the implication of not doing it and how that can really interfere with reproducible results or invalidate all the work that you've done. So if you think you're working on a lung cell and it turns out to be a kidney cell, all of the research is invalidated because it's not the cell line, the cell model.
I do not know why labs are so reluctant to authenticate their cell lines and report their findings. I think perhaps many labs really like particular cell line models that they're using. Perhaps they have a phenotype that goes along with their favorite hypothesis. But it's really a disservice to science if you're not using the right models.
There's such a rush to publish, there's such a pressure for productivity. And then the other two factors, one is complacency. Oh, it will never happen to me. The cell line that I'm working with it's not going to be contaminated. I have great cell culture technique. The other piece that I found the most interesting was that, especially researchers that had been in the field for a while, that were established, there was some reluctance to actually explore the authenticity because they might find something that would invalidate their livelihood, the mark that they made on science if they ever found out that something was amiss. And so it was kind of out of fear.
The cost that's being applied to misidentified cell lines is about $1.3 billion. We calculated that if you could reduce the misidentification rate, then you would lower that $1.3 billion cost to about $200 million.
While contamination and misidentified cell lines are not a new issue in biomedical research, studies over the last 20 years indicate the frequency of cross-contamination in cell lines has reached as high as 46%.
There's a breast cell line that have been used for thyroid cancer and for uveal melanoma, but it turns out they were not derived from a female, as some suspected, but rather from a male that had melanoma. So they're using the cell lines incorrectly. I have a picture here which has an illustration of the problem. Here on your left, cell lines that have been used as a breast cell line. Here on this side are cell lines that have been used as a thyroid model. And down here they've been used as ocular melanoma. And they're all from the skin, from the buttocks of a male. This particular sample shows that it has two X chromosomes and a part of the Y chromosome. And the Y chromosome has been kept in the cell line by being translocated to chromosome 22. So part of it's been rescued, part of it's been lost. That's why people thought it was a female cell line all the time.
The costs and implications of contaminated and misidentified cell lines first garnered attention more than 50 years ago. In 1966, Dr. Stanley Gartler reported that he had found HeLa cells in 18 of 18 cell lines previously thought to be unique. He reported his findings at the Second Decennial Review Conference on Cell Tissue and Organ Culture to a skeptical audience.
It's very easy if you take a cell like HeLa, which grows like mad, and you're working with a cell which doesn't have those properties at all. You make just a teeny mistake and it'll lead to that contamination. So it's very easy for people to make mistakes And once you got one HeLa cell with thousands or hundreds of thousands of others, it would take over because it would grow forever. Most cell cultures wouldn't. There was nobody keeping track of how often this was reported. And I still remember when I was just a few days before I was leaving to go to this meeting on cell culture back east, Herschel Roman, who was a chairman of our department, he said, "You're going to go back to this meeting where all these people are who've worked in cell culture, tell them that they've been working on something that they didn't know what they were working on. It was a contamination. They'll laugh you out of the place." Most people said, "Oh, this guy's crazy." And so I had to really spend a lot of time explaining what I meant by a marker, how we could distinguish it.
More than 50 years later, frustrated with the breadth of the problem, Dr. Roland Nardone wrote to Mike Levitt, the then Secretary of Health and Human Services. The letter pled for action. It called for the requirement of a cell line authentication standard by federal granting agencies. 19 cell culture experts signed the document.
It was my belief that we had to have the support of the NIH if we were to have a worldwide impact on this matter of use of misidentified cell lines. I assembled a group of people who had national reputations. We laid out a plan for the content of the letter regarding the growing problem. I proposed that the NIH and other granting agencies establish a zero-tolerance policy. That would indicate that if an investigator was not willing to authenticate the cell lines that were being used, that investigator would not be supported. No authentication, no grant. The response was very slow. There was no policy approval approaching that of zero tolerance.
In 2016, the NIH amended its grant application instructions to include the authentication of key biological and chemical resources. Similarly, many journals have begun to also require authentication for publication. One of the earliest adopters of this practice was the International Journal of Cancer, which began recommending authentication for publication in 2010. By 2012, the journal required a certificate of authentication for submission.
We have a problem in terms of reproducibility of research results in cancer research. Though at that time we had actually written to many major journals, general scientific journals, but also cell biology and tumor journals, that we should do a concerted action and introduce measures, quality measures, demands that we as journals would have to install. At that time, unfortunately, we got little or no response of the community, and so we started eventually, after we had a certain period of voluntarily doing cell line authentication by the authors. We got also not very much response. We started to make this an obligatory parameter when submitting to our journal.
The current industry standard for cell line authentication is a process known as short tandem repeat analysis.
The advantage of STRs is that you only have to amplify about 15 loci, because each locus has anywhere between 5 to 100 different variants at that locus. So you get a lot of information from one amplification, and the ability to discriminate between different cells just on the basis of 15 tests.
You don't even have to know how to spell STR to get your cell line authenticated, because there are lots of organizations out there that will do it for you at cost that's less than what you're spending every month to maintain the cell line.
So ATCC has been the leader in STR profiling and cell line authentication starting in 1986. And we do offer a service where customers send in their sample. We perform STR testing, STR profiling under 17025. That's an ISO standard. That is a controlled process. Our STR profile report that we do provide to the customers does meet the requirements for the journals, as well as meeting the requirements for the funding agencies, that both of which are now requiring evidence of authentication of cell lines and reagents.
STR profiling is a PCR amplification-based method, but there are other methods that are being developed for other cell lines.
Another way which is possible, it's a little bit more complicated, but it's actually using next-gen sequencing. Next-gen sequencing can look at STRs as well. There's a little bit, of some issues with that, but that can be done. But you can also generate SNP profiles, which are unique to individuals, by using next-gen sequencing as well. Of course, that requires a much longer workflow and much more data analysis than just using STR methodologies.
Next-gen sequencing is the way to go if you've got the moolah, because it's an expensive technology, but you can get many SNPs, you can get many STRs, and the advantage of this plethora of data is that you can see differences that happen between sublines. They're all from the same patient, but they have evolved in culture.
The International Cell Line Authentication Committee, a voluntary group of cell culture experts, was formed in 2012. Since its founding, ICLAC has aimed to make cell line misidentification more visible and to promote awareness and authentication testing as effective ways to combat the problem. Amanda Capes-Davis, the current chair of the organization, says while there will never be a way to totally eliminate misidentified or contaminated cell lines in biomedical research, there are many ways to minimize the risk. In addition to extensive training on the issue, Capes-Davis says journals, funders, and institutions need to take the issue more seriously. Testing early and often is an obvious solution, but beyond good cell culture practice, the industry needs to do a better job of materials management. In the meantime, resources such as the ICLAC website, which lists nearly 500 known misidentified cell lines, researchers also can access information on over 100,000 cell lines through the Cellosaurus database.
Cellosaurus is available online through a website on Expasia, or people can download it. So what is included includes almost every, or at least it tries to be complete in terms of all of the different cell lines which are used in laboratories since the 1950s, when the first cell lines were established. Currently it contains about 100,000 cell lines, and there is a wealth of information around those cell lines, which type of tissue, which type of donor, is it male or female, which type of species it is. So why should scientists use it? Because in fact it's for them a reference in terms of finding some information on cell line and also the publication associated with the cell lines, so they can go back and look at who established that cell line, how it was established, and so on. Anyone can use it. It's freely accessible, so there is no login and no paywall, so it's free of use. And it's basically becoming an ongoing process to contact cell line collection, to feedback, for asking questions, for errors, and so on. And it's growing both in scope and in feedback of users.
Traditionally they really have not had a consistent nomenclature. Now ICLAC has guidelines for how to name new cell lines that you generate, so hopefully if labs adopt that best practice, that at least the nomenclature of cell lines will become more straightforward.
Experts say another solution to the problem could be a better emphasis on institutional memory.
The idea of an institutional memory is to consolidate those cell lines all into one single cell bank, and then generate all the data on those cell lines and store it in a central repository which is linked to those cell lines. So whether it be cell-based assays, whether it be cell-killing assays, whether it be RNA-seq, next-gen sequencing, all of those things can be directly associated. And then those groups within the research organization can then delve in and go back to the cell lines, and hopefully they'll be able to reproduce the data much more rigorously than was able if you were just saying, well, can I have your cell line that was in your bank because mine didn't seem to work? And so the overall effect of that is to save the company money, because you only necessarily have to do your RNA-seq a couple of times, and then it's stored, and then you can go back to that original sample instead of then buying a fresh sample or looking from a sample from another lab, which may not absolutely match.
We also have to dispel these beliefs that it's not a problem in my lab. It could very well be a problem in your lab. And I really think that there are good solutions out there. There are some myths that we have to overcome, that it's simply too expensive, that it's going to slow down their research. It's simply not true. And we also need to develop new methods for authenticating non-human cell lines, because the problem's not just for human cell lines, it's non-human cell lines. It goes beyond cell line authentication to other key critical biological components. We've got to make sure we know what we're using, because the impacts on the science, on the credibility of the science, on the credibility of how we're spending our research dollars, all comes into play.
As a principal investigator, I have graduate students in my lab who I train on cell line authentication and best practices. I also teach an ethics course for the university, which is required by all trainees who are on NIH fellowships, for example. And as a part of that, I teach a lecture on cell line authentication, not only the importance, the consequences, and how it should be done correctly and on a routine basis.
Quarantine the cell line when you get it in and check that it is what it is by profiling. At least then you know what you're starting with. Then a simple thing that you can do is that when you're doing passaging, take an aliquot, it's usually by dilution, so you have some media with suspension of cell left over, put it in a Eppendorf tube, spin it down, freeze it at -20. You're not concerned about survival. All you want is saving the DNA. Take that once a week or once a month. Then when you're finished, do a profiling of it. If they don't match, then you have a set of points in there that you can check by profiling. Where did the problem arise? Was it just at the end that it got switched, or was it somewhere at the beginning or in the middle of your experiment? So I urge people to at least collect the samples, profile before you start working with it, during and after, before you submit.
Now the NIH does require a statement on how you will authenticate cell lines. So I always include that information in my grants. I know smaller associations are also doing that. I am a member of the American Thyroid Association. Now we require cell line authentication for our grants. And I think it could be, more rigorous in that you need to spell out more, precisely what exactly you will do to authenticate and how often, but hopefully that'll come with time.
As a community, we need a unified strategy. I don't think any one stakeholder bears the brunt of the issues that we have. I think education is absolutely necessary. I think the old folks like me are less likely to learn than the new kids coming through. And part of the problem is, some of the people who are mentors are not necessarily well trained themselves. So they're perpetuating poor training. So a good training program, institutes, both academic and industry, need to have training programs which raise awareness for this.